Amar Kumar is a PhD student in electrical and computer engineering at McGill. He also works under the guidance of Professor Tal Arbier in the probabilistic vision group. Amar completed his undergraduate in the Department of Engineering Physics at IIT Delhi. Additionally, he completed his master's in electrical and computer engineering at McGill under the supervision of David Plant. Amar's primary research is in the field of neuroimaging analysis and machine learning. Currently, he works on using deep learning methods to study the temporal progression of multiple sclerosis. The floor is yours.
No, right. Hello, everyone, and thank you to Unique for providing me with this opportunity to present my research work. So today I'll be talking about explainable machine learning models in medical imaging, and let's jump right into it. So we have seen this deep learning as a growing field in various medical imaging research areas, and here are few examples. The list is non-exhaustive, and that would take a lot of slides to include all the work. So I'll just go through a couple of directions where deep learning has proven to be useful for medical imaging. So on the box that you see in purple color, you have a mammographic mass classification, which is trying to classify mammograms based on the images that are detected in scans. The second is segmentation of lesions from the brain, which is also done using deep learning models. And those red areas that you see in the brain, those are the lesions which are automatically detected by a deep learning model. The next one is breast cancer metastasis detection. So again, another field where deep learning has proven to be very useful in detecting these breast cancer cells. That being said, we have seen a huge gap that these models are not widely applicable in clinic, and we'd ideally want to know the reason why haven't they been integrated into clinical workflow? Are there any shortcomings? Even though if some of you have worked with these models, you will see that some of them have great accuracy of something like 85, 90%, or even more than that. So what's the problem in including them in clinical workflows? So we'll talk a bit about that. So there are a couple of challenges associated with deep learning in clinic. The first one being that these are very large scale images and there is lack of relevant training data. So it's not only that you get the scans and they are as good as they can be used for deep learning models. They need to be properly segmented. You need to know the ground truth. You need to have baselines such that you are able to compare these models to human accuracy, which means that clinicians working in those specific field need to provide information so that deep learning models can learn and train on them. Also, these data on which the models are developed, you are unable to build robust or generalizable models, which finally forces these deep learning models to make potentially deadly mistakes. And I'll focus on some of the example. And finally, these deep learning models have somehow become like a black box. So we provide certain input and we get an output, but we don't know what's happening in between. So my research in Professor Arbil's lab is focusing on that part, which is developing explainable AI models, trying to know the reason why a model makes a specific decision. So in the end, it's about deep learning models for medical image analysis requires trustworthiness. There has to be some sort of bridging the gap between the clinicians and the patients itself, so that they are able to explain that if a deep learning model is predicting in a certain way, why are those predictions made? So here are a couple of examples where deep learning models can make potentially deadly mistakes, which was we all saw a couple of years ago, not very far, that COVID happened and hundreds of AI tool was developed, but they failed to catch COVID or detect COVID, and none of them widely helped in detection. There is a reference provided at the base of slides. So if you're interested, once these slides are shared, you can also take a look if you're interested in these areas. In addition to this, deep learning model can be biased as well. Here are two different examples. On the left, you have gender imbalance in medical imaging, producing classifiers that are biased. So these classifiers can be biased in such a way that they perform well on patients who are male, but do not perform well on the patients who are female, or the other way around, they perform well on female patients, but do not perform well on the male patients, which is a bias that can be induced in the model. The second kind of bias can happen in an area. Imagine that you're trying to detect skin cancer, and based on skin color, these deep learning models can induce a bias. So they are detecting cancer in one skin color pretty accurately and are not detecting these cells in other areas pretty well. So there is a need to mitigate the bias as well. And finally, just to enforce this point that I'm trying to make, this is a screenshot, that picture that was taken at McGill, which is Medical Image Computing and Computer-Aided Intervention, one of the conferences that is held to promote deep learning in medical imaging. And if you can look at the quote, it's that the challenge to radiologists embracing AI in practice is that we don't really understand how AI arrives at a particular decision. So all this motivation is to focus on one single point, that there is a need to open up the black box. Hence, the overview of this talk is to focus on explainability and discovery of predictive image markers, which is focused towards trustworthiness and reliability of deep learning models. The other aspect of developing trustworthiness is improving fairness, but I'm not going to go into the details of improving fairness. And the case study that I have used is multiple sclerosis as well. So if we talk about multiple sclerosis and the amount of data we have first federated clinical MRI data set, which is really large, and we are lucky and thankful to our collaborators and sponsors who were able to provide us with this data. So we have tens of thousands of patient scans from multiple global imaging center and on multiple scanners, which means that we have federated data from different scanners around the world for different patients, as well as we have large MRI volumes, which means that the resolution at which we can look at images is quite high. And on the left, you have a couple of images showing different modality, and each of these modality have different physics involved. So they capture different things that is associated with the lesion. And this might seem like a deja vu for what my colleague already presented. The slide is important because I wanted to stress on the fact that it's really important to discover biomarkers or imaging markers, very specifically in this case, such that you can do an early detection of multiple sclerosis. So imagine a scenario when a patient is coming in, and with some confidence, the doctor is able to say that I'm probably 50 or 70% sure that your situation might worsen in future. And that's a very helpful thing to have in clinic, because if you are able to discover these biomarkers, or let's say fingerprint of the disease before disease can actually even happen, that's a wide use case in clinical scenarios. So I'll show you one of the research that we published from our lab. And hence, this entire talk is about trustworthiness of image-based personalized medicine for explainability, which is opening up the black box. So this is the scenario which I was just explaining, is a patient is looking at the image and a clinician is looking at the image and say that I can't tell how this model decided, or I can't trust the model. Also, trust is one aspect. The second aspect is to say that whether this person will be sick in future or not. Essentially, one of the ways in which this is done in medical imaging is using GradCam, which is gradient-based class activation maps. So using a deep learning model, you try to highlight which regions are important in order to make certain predictions. And this is what a GradCam looks like, which is shown in the image over here. If you can see my cursor, and the areas that are shown red are the important areas. But there is a missing piece of puzzle, which is GradCam simply is focusing on the area which is answering the question like, where was the model looking at when it made the prediction? But it's not telling us what exactly is the model looking at. It's just a heat map that is shown on top of image. So ideally, we would want to answer this question, that what are patient-specific image markers that are predictive of future responses? And in order to answer this, we we provided a method which was explainability via counterfactual synthesis. So what a counterfactual is simply answer this question that how the patient's current or the baseline image look were it to have a few different future disease outcome. So whatever happens in an actual scenario, imagine another hypothetical scenario such that few things were absent in this patient's brain. And the absence of those things indicate that this person will behave differently in the future. So essentially, this is the usual case scenario where you have a baseline image and all the lesions that are detected in this baseline image are shown in those red outlines. And what you see is year one. So after a year, the same patient was scanned and you saw that there was 2 new lesions that are developed over here. So in the green and in the blue outline. So focus on those areas. So ideally, what we want to do is what should change in the baseline image such that I don't see those new or enlarging lesions. So essentially, we'll be using a generative AI model to have this complete pipeline. So the bottom row is an alternate scenario where the baseline is changed using a generative AI technique such that we have a counterfactual image which has a different disease outcome so that these crosses that you see, those crosses are put over a lesion so that you won't see a future lesion that's going to happen to the same patient. So essentially, if this is done in a perfect scenario, what you will be able to do is analyze or come to a conclusion that these areas are really important for this specific patient, because if these areas were focused at, this person would not be sick in future. So that also promotes patient-specific drug discovery, targeted drugs, so that you can target those specific areas. And here is a complete analysis, which is identification of personalized markers for future patient outcome. So you see 4 different images, and I'll go through each of them a bit slowly because that might be a lot to take in. So on the very left, you have the real image, which is the actual patient along with lesions shown in these red outlines. The second image is a counterfactual image, which is synthesized using the research that I just showed. These counterfactual images also have few lesions. I don't know if it is visible on the screen, but if you can follow my cursor, these are the lesions that are being shown. So ideally, what happened between real and counterfactual image is that a lot of lesions were suppressed. And the suppression of these lesions indicated that this patient will not be sick in future or will not show future disease activity. And if I take a difference map between these real and counterfactual image, these difference maps highlight these specific areas. Now, what's interesting is if you look at these periventricular regions, these areas correspond to the same areas where the patient was having new or enlarging lesions. So these areas correspond to this region, but the model also is suggesting that you need to focus on these areas as well, which is interesting because nothing happened in future in those regions, but this disease is heterogeneous and we need a lot of validation techniques before we can claim that we have discovered the so-called new biomarkers. But this is a preliminary approach that we showed works for the case of MS patient, and going into a bit of detail about the counterfactual synthesis model. This is very specific to deep learning. So what we are doing over here is these counterfactual images are put under certain constraint. And these constraints are 3 specific things, which is subject fidelity. It should look like the images belong to the same subject. So your generative model should not simply copy any other image that is out there in your training sample and say that, hey, this is a non-sick person. So you take this, you try to maintain the subject fidelity. The second one is you try to maintain that your counterfactual image belongs to the correct target class, which means that non-sick class or the healthy class. And third is realistic images. It should not pop up a person's image that does not have MS, because that will be a null scenario that your generative model is saying, hey, if this patient did not have any lesions at all, it would not be sick. Well, that's true, but it's not true in an ideal world, because usually even people who do not have MS have lesions in their brain. So That being said, these are the part of the network. If you're interested, I'm happy to talk more about that. But these are the part of network that focuses on subject fidelity, which is the generator that we are trying to use. So an input image goes through a generative model, which tries to create two things. One is the reconstruction image, which is trying to know model's capacity, how well it can reconstruct an image. And second is the counterfactual image, which is what we are interested in. The second one is a pre-trained classifier that's embedded into the network, which tries to maintain that the counterfactual image belongs to the correct target class. And finally, the last is a discriminator, which is a very popular approach in generative-based synthesis network, where you have a discriminator that checks the realism of the image, which makes sure that the images that are generated from the network are in fact realistic. So essentially, that's all about the details of the work that we submitted. So there is this final question. All this looks great and happy, but are we ready for clinical deployment? And I'm not sure, but this is what the promise of AI for clinical decision support would ideally look like. So you have a healthcare patient and you have some data that is extracted out of it. This is where most of our research sits in, which is trying to develop AI techniques such that you can build a trustworthy AI system that can be incorporated into clinic. And that decision-based system mainly comes from the domain knowledge that you have and also the clinician's support. So with that, I would like to thank you all for your attention. I would also like to thank my co-authors for this paper, as well as our sponsors who generously supported us in this research. So thank you, and I'm happy to take any questions.
Hi, thank you for the talk. It's more of a curiosity questions. So if I will understand what you say, one problem by using AI actually is that they do errors. And of course, we don't want them to do errors. And I'm wondering actually compared to humans, how much if they do more errors than humans, or if they do different errors, or because I mean I guess also actual humans do errors. So how does it compare? Then is it bad finally, or is it just an help? I mean, you see what I mean? Like a clinician doing every day at the end of the day also do errors. So I'm just wondering how it compares.
That's a very relevant question, and in fact, a question that's being debated in the field as well for quite a long time. That even humans make error. Humans make error. So why is it that deep learning models that is also making error? How do these two errors compare to each other? From my naive knowledge and understanding, I would say that yes, deep learning models will also make error, but the human error is restricted to the data that they have seen, and the field such as deep learning, where they are able to federate a large amount of data, it's definitely more than what a single human will have experienced in their lifetime. So what we are trying to do is this federated based approach. We have lots and lots of data and assigning different modeling techniques, which is one which I focused on my talk, which was about explainability, but there are other aspects as well, which my colleague talked about, which was uncertainty. So even though you are making an error, that's okay. But how sure you are that you are actually making an error versus when you're not making an error, how sure you are that you're not making an error. So trying to know the bounds, which is an uncertainty estimation associated or in collaboration with the explainability, I think is the way to go. But the last point that I would like to make, which I didn't focus on my talk, but was mentioned in one of the slides, is you also need to understand that even though there will be large federated amount of data, there will be biases associated with each of these data. And in order to exploit or at least be understanding towards these biases is also something that needs to be considered before these models are actually deployed into the clinic. Thank you.
Thank you for the lovely talk, Amar. So we will now have a 15-minute coffee break. Then we will return for a panel discussion on synergy between industry and academia.